Okay, so in this lecture we are going to discuss how fats are oxidized. So here we're going to learn again once more how fats are going to be a major fuel source. And this is going to be important especially after fasting. And we're going to talk about the different fats, and then we're going to discuss beta oxidation of your fatty acids. So your adipose tissue is going to release fatty acids, and this is going to be triggered mostly by hormones. Glucagon, epinephrine. Glucagon is fasting state. Epinephrine is the fight or flight hormone. And this is going to stimulate adipose tissue via activation of two enzymes. One is the adipose triglyceride lipase, and that's going to help to release fatty acids. And you can see right here the word triglyceride. And so it's going to work on your tags when you have 3 fatty acids present. You're going to need for your adipose triglyceride lipase, you're going to need a co-activator. And this is referred to as your alpha beta hydrolase domain containing 5, or ABHD5. And ABHD5 is basically going to keep your adipose triglyceride lipase, ATGL, away from fat until it's activated, and it's going to have some help in doing that as well. So there are 3 lipases that are required to release tags. You have your adipose triglyceride lipase, you have your hormone-sensitive lipase, and then you have your monoglyceride lipase. So both of these have sort of an indicator of what their substrate is. Your tri is for when you have 3 fatty acids on your glycerol. Your mono is when you have 1 fatty acid on your glycerol. And then, of course, your hormone-sensitive lipase is right in between, and so that is when you have 2 fatty acids on your tag, or your glycerol. So just really fast, you have some sort of hormone signal, right? And then you get your normal signal transduction. And then you're going to get your perilipin here. And so what perilipin is perilipin is sort of a docking place. And your perilipin is going to hold that cofactor of your triacyl glycerol lipase. And so that ABHD is going to be docked on perilipin, and perilipin is associated with a fat droplet. Now, what happens to it is signal transduction happens, and of course, you're going to activate kinases when you have your epinephrine or your glucagon. And so these kinases are going to phosphorylate the perilipin, which is going to release your cofactor. So you get your kinases, and then your cofactor is released. And then the cofactor can help that first enzyme take off 1 fatty acid from your tag. And now you end up with a diglyceride. And then the hormone-sensitive lipase will also activate your hormone kinase will also activate your hormone-sensitive lipase. And then you get another fatty acid removed, and then you're going to end up with a monoglyceride. Now, monoglyceride, the lipase for monoglycerides is constitutive. So that means it's always on. So as soon as you have a monoglyceride, it's going to pick up on that monoglyceride, remove that 3rd fatty acid, and release glycerol. All right, so now we released our fatty acids. They're going to be released from the adipose tissue. Sorry, this got kind of cut off. And so they're transported through the blood via serum albumin. And so remember, these fats, these monoacyl, are going to be very hydrophobic. And so they bind to serum albumin, and serum albumin will transport them through the body. And then they're going to be picked up by cells. So in the plasma membrane of cells, you're going to have receptors that will pick up fat. They're going to be taken up by the cells, and then they're going to be activated in the cytosol. And the activation is by adding an acyl group, an acetyl-CoA. So just like we had activated sugars when we added UDP, when we add that coenzyme A, we have activated fat. Now, the activated fat is going to be able to get transported through the outer membrane, the mitochondria, and then it has to be added to a carrier, and the carrier is carnitine, and then carnitine is going to pick up the fat and carry it through the carnitine carrier. Once inside the inner membrane of the mitochondria, the carnitine is removed, and you get, again, your fatty acyl-CoA. And then fatty acyl-CoA is going to go through beta oxidation, and you're going to generate acetyl-CoA, and we know what acetyl-CoA does. All right, so let's just break this down and go over this again. We're going to talk about a few more things that we're going to add. So again, we released fatty acids from your adipose tissue. So these are free fatty acids plus glycerol. And then the free fatty acids are going to bind to serum albumin. And they're going to be picked up by cells. Your glycerol, of course, is going to go to the liver for gluconeogenesis. Okay, and so you're going to have different proteins on the surface of the cells to pick up your fats. They fall into different categories. We have a translocase, we have binding proteins, transport proteins. It's just basically cells have ways to pick up fat because fat are obviously going to be a very important source of energy during the fasting stage. All right, so now let's talk about how we get that activated fat. Important, activation must occur before beta oxidation can even start. And one of the reasons is because you have to be able to get this into the mitochondria matrix. And so it all has to do with the molecule, how it looks, what's it binding to, and the transporters. Now, the activation is going to be catalyzed by a fatty acyl-CoA synthase. And this, of course, is going to, again, be similar. It's going to depend on the size of the fatty acid. And so the fatty acid will come in, and you're going to have the fatty acid synthetase, and it's going to add to it a phosphate group. And so you're going to get a phosphate group with adenosine attached to it, and you're going to release a pyrophosphate, which is a very good leaving group. And then you're going to add a CoA. So your CoA is going to come in, replace or disperse the AMP, the adenosine monophosphate, and then your fatty acid is now activated with the coenzyme A attached to it. So it becomes a fatty acyl-CoA. Now what happens to these fatty acyl-CoAs? Well, just like our sugars, once you get the sugar in, you can go in many directions, and the same thing with a fat. So once you get the fat in and once it's activated, it can go in many directions. You can make energy, which is what we're going to be talking about through beta oxidation. You can make membrane lipids, or you can make tags and send it back out to be stored. So all of this is going to depend on the needs of the actual cell. So if you're going to synthesize your triacylglycerol and your phospholipids, this is going to occur in the endoplasmic reticulum. If you're going to oxidize and have plasmalogen synthesis, this is going to occur in peroxisomes as well. It can occur also in the ER we'll talk about. Beta oxidation only occurs in the mitochondria, and we'll discuss that as well. It also occurs in the peroxisomes to a certain extent. Okay, so now that we have our activated and we know that we need energy in the cell, so we want to get this activated fat into the mitochondria, we're going to have to transport it. So again, we had our fatty acid, we had our acyl-CoA synthetase, we brought in an ATP and a coenzyme A, and we spit out the fatty acyl-CoA, our activated fat. Our activated fat now enters the inner membrane of the mitochondria, and within the inner membrane of the mitochondria, you're going to exchange your coenzyme A, remember it's the activated portion, for fatty acylcarnitin. And the fatty acylcarnitin will enter the inner membrane or the matrix of the mitochondria, one-to-one exchange for carnitin or carnitine. And then so you're bringing in a fatty acylcarnitine, you're leaving a carnitine, which can be then used to pick up another fatty group. Now, once you're inside, and this is carried out by a transferase, carnitine palmitoyl transferase 1, which is on the outer or embedded in the outer membrane. On the inner membrane, you have another enzyme, which is transferase 2. And so this is basically going to exchange CoA for carnitin, and so you get your fatty acyl-CoA remade, and you release your free carnitin. And so it's important just to point out that transferase 1 is exchanging, bringing in a carnitin and exchanging that for CoA, so it releases a coenzyme A. And transferase 2 picks up a CoA and uses it to reactivate. All right, so now we're in the mitochondria matrix, and so we have all the enzymes there that can do many different things, including make ATP. But we have to get the fat in a form that these enzymes can use. And so all of this is to get the fat into the form of acetyl-CoA so that it can enter this TCA cycle or citric acid cycle. And then, of course, those carbons can be fed through the TCA cycle and generate energy through the electron transport chain. Now, when you're doing beta oxidation, we're also going to generate some energy through NAD and FAD, and we'll discuss that. Now, the stages of fatty acid oxidation to get to energy, stage 1 is our beta oxidation. Stage 2 is acetyl-CoAs go through TCA cycle, and then stage 3 is the electron transport chain. So for beta oxidation to occur, we're going to have 4 steps, and they're going to repeat over and over again. The first step is going to be oxidation, and we're going to have FAD pick up electrons in this step. The second step is going to be a hydratase. The third step is going to be another dehydrogenase. In this case, we're going to have NAD, so we're going to make an NADH. And so just like when we discussed the TCA cycle, now we're in the mitochondria matrix, so if you generate an NADH, you're going to be able to donate those electrons to that enzyme 1 of the electron transport chain, and you're going to be able to make a lot of energy. FAD, on the other hand, cannot go and donate its electrons. So we'll talk about how FAD is going to transfer electrons to, as you could probably guess, coenzyme Q. And then finally, you're going to have a thiolase, and the thiolase is going to generate your acetyl-CoA, which then can enter the TCA cycle. So we'll just go through these steps, and I'll just point out things that are important. So here we have the first step. You're going to have a dehydration step. This is going to be an acyl-CoA dehydrogenase. Depending on the size of the carbons, you're going to have different acyl-CoA dehydrogenases. So that's just a very general term. They're all going to be associated with FAD, and so you're going to get some electron storage in that way. And then you're just going to generate a trans delta 2 acyl-CoA. And so this is just the next step, and the enzyme next is going to be specific for that acyl-CoA, whatever size carbon that happens to be. Now, just as a side note, we have very long chain acyl-CoA dehydrogenases. We have medium chain acyl-CoA dehydrogenases and small chain acyl-CoA dehydrogenases. The second step is going to be hydration. We're going to add water, and this is through a hydratase. And this is basically going to generate an L-beta hydroxy acyl-CoA. So you're basically just breaking that double bond. And then you're going to have the second, or the third step is going to have the second dehydrogenase. This is a beta hydroxyacyl CoA dehydrogenase. Again, it's going to be specific for the length of carbons. This is going to be using NAD as its cofactor, and you're going to generate NADH. You're also going to generate the beta ketoacyl CoA. And so you can see what's happening here is we're generating this leaving group, and then we're adding this oxygen so that you can make another acetyl-CoA afterwards. And so we are in the cell, we are in the mitomatrix. And what does NADH transfer electrons to? It's to enzyme 1 of the electron transport chain. Now, the fourth step to generate acetyl-CoA is going to be a thiolase, an acetyltransferase. And you're going to need a second mole of coenzyme A, and this is to add to the long chain of fatty acids that are still there. And you're going to have an acetyl-CoA as a leaving group. And so we went from a carbon-16 to a carbon-14, and carbon-14 is a mistral-CoA. That's just the name that it's given. And then the carbon-14 will go through the process again, and it'll become a carbon-12, and then a carbon-10, and so on. So these 4 steps are going to repeat over and over again. You're going to have the cleavage of the last 4 carbon acetyl-CoA. This is butyryl-CoA. And that last step is going to generate 2 acetyl-CoAs. And so if you have an even chain fatty acid, such as a palmitoyl-CoA, which is a common fatty acid that your body makes in the liver, it's going to get cleaved 7 times. And in that 7 times, it's going to produce all of these. It's going to produce 7 FAD, 7 NADH, and 8 acetyl-CoAs. Now, when we talk about your FADs, again, your FAD is attached strongly to the enzyme, so it cannot directly donate its electrons to the electron transport chain. And so you're going to have a series of transfers. So you're going to have that acetyl-CoA dehydrogenase pick up the electrons, and then it's going to transfer it to an electron transferring flavoprotein, and then that's going to transfer it to coenzyme Q reductase, and then you're going to get coenzyme Q that will take those electrons in the electron transport chain and donate them to enzyme 3 of the electron transport chain. So how much energy do you get out of beta oxidation? So you get a lot. You can imagine you have 1 mole of your 16 carbon, which is palmitate. You get 8 acetyl-CoAs. Those will all feed through the TCA cycle. You get 7 NADHs. Each of them will give you 2.5 ATP, so that's 17.5 ATP. 7 FAD, 1.5 ATP each, and so you get 10.5 ATP. If all acetyl-CoAs go through the TCA, you're going to get 8 GTP, 24 NADH, 8 FAD, which overall you're going to get about 80 ATP. And so this is a way that your liver gets enough energy in order to do things like gluconeogenesis. So this is just a table to show you that there are many different acyl-CoA synthetases, different acetyltransferases, and they all have to do with the size of the fat that they're working on. And so you have your medium chain, which is your octanol carnitine transferase. You have your long chain, very long chain synthases, as well as medium chain. And so they're going to be present in many different things. You don't have to memorize all these, but just understand that if you see medium chain acetyl dehydrogenase is working on a medium chain fatty acid, so about 6 to 8. Small chain is going to be working on a 2 to 4 carbon fatty acid. Okay, so here we talked about how fatty acids are major fuels. We also discussed our beta oxidation and what you're making on the beta oxidation. We talked about how fatty acids are released from the adipose tissue, how they're activated, and then transported in the mitochondria. Now, as you can imagine, NADH and acetyl CoA are going to be important, and they're going to be important in regulating this whole process.